Hi everyone, my name is Alejandro Paredes and I am a lecturer in pharmaceutical sciences in the School of Pharmacy at Queen's University Belfast. The School of Pharmacy is a center of reference in research and education in pharmacy and pharmaceutical sciences. And in this presentation, I will show you some of the work that we have been doing in our labs on the use of nanocrystals for controlled drug delivery. The presentation will be divided into four main parts. At the beginning, we will discuss the problem of poor solubility of drugs in the pharmaceutical industry and how nanocrystals can help us to overcome this issue. Then we will take the example of albendazole, which is a very hydrophobic molecule that is used for the treatment of echinococcosis in humans. And then I will show you how the nanocrystals and microneedle technology can be combined to deliver drugs locally and systemically. And then we will have some final remarks. The issue of poor water solubility is very well known among pharmacists working in research, both in the academy and also in the pharmaceutical industry. We know that 90% of novel chemical entities, up to 60% of drug candidates and 40% of already approved drugs are poorly soluble in water. Historically, several strategies have been used to increase the dissolution rate of these drugs. For example, the use of cosolvents, cyclodextrins, solid dispersions, oil-water emulsions, and the preparation of co-crystals. However, these strategies are limited to physicochemical characteristics of the drug. And this is also reflected by the low number of products based on these strategies that are approved in the market. The reduction of particle size is also a very attractive approach that allows to increase the specific surface and increase the dissolution rate of solids. So initially, the particle sizes of micronized powders were between 2 to 10 micrometers, but then with the development of novel technologies, the particle size could be reduced to the nanometer range, and then the nanocrystals were developed. Nanocrystals are in essence nanoparticles made of the drug, and they have crystalline properties. Their particle sizes are usually between 200 and 800 nanometers. Because they have an enlarged specific surface, they have an increased saturation solubility, dissolution rate, and mucoadhesion. And this is usually linked to an increased bioavailability. There are more than 20 products already approved in the market, and most of them are used for oral administration of poorly soluble drugs. When we compare nanocrystals to other nanoparticle-based drug delivery systems, like solid lipid nanoparticles, polymeric nanoparticles, liposomes, nanogels, for example, nanocrystals have a series of distinctive advantages. They can be easily produced, they have long-term stability, solvent-free manufacture, scalable production, neutral pH, feasible sterilization, and more importantly, they have a greater drug loading as they don't need to use carrier materials. They are made up to 100% of drug. Human echinococcosis or hydatid cyst is one of the diseases in which we have a particular interest and in which we have been working during the last years. It is a neglected disease and it is caused by a parasite of the genus Echinococcus. There are currently 1 million people living with the infection, and only in 2015, which is the latest statistics available, nearly 20,000 people died because of this disease. It's distributed globally and it is endemic in some areas, and usually it is related to rural life and conditions of poverty. It is a difficult to treat disease because it requires surgical procedures that are often not available or inexistent in the areas where the disease is prevalent. So the treatment of preference is usually the use of antiparasitic treatments. If the cysts are located in undangerous places of the body, the approach could be just to watch and wait. Albendazole is a commonly drug used for the treatment of human echinococcosis. Despite the drug is very active against the parasite, it has a very poor solubility in water, which produces an erratic bioavailability and frequently a lack of efficacy. Albendazole is administered in the form of tablets that must disintegrate in the stomach, where the drug is dissolved and then absorbed in the first part of the duodenum. Albendazole is completely metabolized to albendazole sulfoxide, the metabolite, the active metabolite of the drug. So to overcome the solubility issues of albendazole, we prepared nanocrystals of the drug using a technique named high pressure homogenization. In this technique, the suspension of the coarse drug is forced to pass through a very thin gap inside the instrument at a very, very high pressure, up to 1,500 bars. So this creates a phenomenon called cavitation, where the drug particles are broken into smaller particles, and with different cycles, the particle size reduces until the nanometer range. Then the nanosuspensions obtained were dried using spray drying technique to obtain redispersible powders. As we can see in the first graph on the left, the application of multiple cycles at 1200 bars produced a reduction in the particle size. So after 30 cycles, the particle size was of around 420 nanometers. We tried different stabilizers here, but Pollock-Summer was able to produce smaller particle sizes and also smaller polydispersity indexes. In the graph on the right, we can compare the particle sizes of the original nanosuspension and the particle sizes of the powders after spray drying when they were dispersed in water. As you can see, nanocrystal 4, the formulation on the right, which contains the highest proportion of Pollock-Summer, were able to redisperse at nearly the same particle size of the original suspension, indicating a high redispersion capacity of these materials. When we observed the bulk albendazole under the microscope, we saw that the particle size was very heterogeneous, and we had particles very small, from 1 to 2 microns, to particles up to 50 microns, like this one here. On the other hand, when the nanocrystals were observed under the microscope, we saw these spherical particles that were formed during the spray drying, but further magnification allowed us to see the nanocrystals embedded in these spherical particles, with sizes of around 400-500 nanometers, as we observed in the previous slide. In this slide, we are going to see what happens when albendazole is in contact with water in comparison to the nanocrystals. So on the left-hand side, we have the beakers containing albendazole, and as you can see, the drug is very, very hydrophobic, and it will be floating on the surface of the water, and it can be like this for days and even for weeks. Whereas on the right-hand side, you can see the nanocrystals being easily redispersed in water. Remember that these nanocrystals were spray dried, and Pollock-Summer was present in the formulation. So the Pollock-Summer forms bridges between the nanocrystals, which are dissolved, allowing the redispersion of the drug nanoparticles. If you imagine this happening into the stomach of a human being or an animal, you could easily imagine that the nanocrystal will be absorbed in a greater extent. And this is exactly what we observed when we studied the dissolution behavior of the nanocrystals and the pure drug. On the left, you can see that the albendazole dissolved only in a 5% after 2 hours of experiment, whereas the nanocrystals were able to produce up to 80% of the drug being dissolved in the same period of time. When the nanocrystals were administered to mice, we saw an increased absorption of the drug, which is reflected in a bigger area under the curve. Also, the Tmax was reduced for albendazole, whereas the Cmax was increased at least twice in comparison to the core suspension. We also evaluated the efficacy of the nanocrystals against the parasite in infected mice. Once the animals developed the cysts, they were treated with an albendazole suspension and also with the nanocrystals. After 30 days of treatment, the cysts were removed from the animals and the efficacy was determined in relation to the weight of the cysts. As you can see, with the nanocrystals, the efficacy was of 76%, and in the picture you can see that the size and the number of cysts are decreased. This was also confirmed by the microscopical analysis of the germinal membrane of the cyst, where we could see that the nanocrystals had a reduced number of cells and also a completely disrupted membrane as observed in the TEM microscopy. We also evaluated the viability of the protoscolysis, which are the early stage parasites inside the cyst, and we could see that the nanocrystals were able to reduce the viability up to 30%. In the images we can see the different protoscolysis in the normal state, and also the group treated with nanocrystals has a completely different morphology and loss of the main structures of the parasite, indicating that the nanocrystals were able to produce more damage on the parasite structures. In this section of the presentation, we are going to see the use of microneedle array patches to deliver nanocrystals through the skin. Microneedle array patches are, in essence, patches that contain little projections that can pierce the stratum corneum and deliver drugs to the viable layers of the skin. Because of the height of these microneedles is lower than 1 millimeter, they cause no pain or bleeding when they are applied to the skin. As you can see in the figures, microneedles can be of different geometries and materials and can be used for drug delivery purposes, but also for absorption of fluids from the skin and patient monitoring. Microneedles can also be self-applied and also used for the delivery of long-acting treatments. So one of the ideas in which we have been working is in the use of microneedles to deliver nanocrystals through the skin. As you remember, albendazole is fully metabolized in the liver, but using this approach we could avoid the first pass and have the parent drug circulating in the plasma. We know from the literature that the parent drug albendazole is more active against the parasite than the albendazole sulfoxide, which is also active. This work was carried out in the laboratory of Professor Ryan Donnelly in the School of Pharmacy, who specializes in the use of microneedles. In this case, we produced nanocrystals using a media milling technique. It's very simple, so we used zirconia beads to produce the milling of the drug particles using magnetic stirring. Here you can see the particle size profiles using different concentrations of poloxamer and twin, and from the results you can see that the crystals have a similar particle size to that observed in the high pressure homogenization of around 450 nanometers. A similar trend was observed for the polydispersity index, where the higher concentration of the polymer was able to produce lower polydispersity indexes. When we evaluated the release profile of the nanocrystals, again we were able to see a higher dissolution rate for the nanocrystals in comparison to the coarse drug. So the nanocrystals were then loaded into dissolving microneedles. To form the microneedles, we use a blend of PVP and PVA, which are water-soluble polymers. We prepared the first layer containing the needles, and then we added a free drug baseplate. This allowed us to obtain a microneedle patch with the nanocrystals contained only in the tip. On the left of this slide, we can see the nanocrystals here in A containing increasing amounts of the coarse drug, and then the microneedles containing increasing amounts of the drug nanocrystals. The microscopic analysis revealed that the microneedles were sharp and fully formed. The mechanical characterization consists of the application of a vertical force to the microneedles against a flat surface. Then we can evaluate the reduction in the microneedle height. It is possible to see that the formulation A was the one with the lowest microneedle height reduction. In a complementary manner, the insertion in the parafilm skin simulant model can help us to predict how well the microneedles will insert in the skin. Complementary OCT experiments demonstrated that the needles were fully inserted in the parafilm layers and also in the skin. The administration of albendazole coarse drug and albendazole nanocrystals orally produced a similar pharmacokinetic profile as what we observed in the mice. Here, the main molecule detected in plasma is the albendazole sulfoxide produced after the metabolization of the parent drug. Of course, the nanocrystals have an increased absorption, which is translated in a bigger area under the curve and a higher Cmax. However, when the same materials, the coarse drug and the nanocrystals, were administered using microneedles, the pharmacokinetic profile is different because now the main molecule detected in the plasma is albendazole, the parent drug, in blue. Again, the nanocrystals were able to produce a higher concentration of the parent drug. The two metabolites, the sulfoxide and the sulfone, were also detected in plasma but at lower levels. Another point to highlight here is that after 72 hours, both systems were able to produce detectable concentrations of the drug in plasma, which tells about the potential for long-acting of these systems. So these results are very encouraging because the parent drug circulating in the plasma could be very beneficial for the treatment of human echinococcosis. In a different work, we used a similar approach with formulating nanocrystals of the drug itraconazole, which is an antifungal molecule, and we loaded the nanocrystals into dissolving microneedles to deliver the drug locally. We used twin PVA and poloxamer 127 as stabilizer, and different concentrations produce similar particle size decrease with similar trends in the size and also in the polydispersity index. Poloxamer 127 was able to produce the lowest polydispersity indexes and particle sizes. So this one, 2% concentration, was selected for this work. In the microscopical images, we can see heterogeneous particles of the itraconazole on the left, but smaller and uniform nanocrystals on the right. After the physicochemical characterization, we evaluated the release of the drug, and we were able to see that the nanocrystals presented a higher dissolution rate, similar to what we observed for albendazole before. The coarse drug, on the other hand, was able to dissolve up to 20% by the end of the experiment. So again, this is a microscopical characterization of the microneedles. We can see that they were fully formed and they have sharp needles. The height of the microneedles was of around 750 micrometers, which is enough to deposit the drug in the skin, but also to avoid pain and bleeding, as we discussed before. In this set of pictures on the right, we can see the microneedle dissolution after they are inserted in water. After 30 minutes, we can see that the microneedles have been completely dissolved. This is a mechanical characterization that refers to the microneedle height reduction. of different formulations containing increasing amount of nanocrystals. Formulation 1 to 4 presented similar results in terms of the reduction of the height, whereas formulation number 5 was not mechanically strong. Similar behavior was observed in the parafilm test, where formulations 1 to 4 were able to pierce up to 4 layers of parafilm. Dermatokinetic experiments revealed that the nanocrystals loaded into the microneedles were able to produce an enhanced drug deposition in the epidermis and in the dermis in comparison to a needle-free patch and a cream, both of them loaded with the nanocrystals. Further experiments in a skin fungal infection model demonstrating that the colony-forming units were significantly decreased for the microneedles loaded with the nanocrystals. So this enhanced capacity of the microneedles loaded with the nanocrystals to depot the drug in the skin was also related with an enhanced efficacy of the drug against the fungal infection. So as final remarks, we can say that nanocrystals have been extensively used for the delivery of poorly soluble drugs through the oral route, and this is because nanocrystals have an increased specific surface, which leads to a better dissolution, absorption in the gastrointestinal tract, and also an enhanced therapeutic response. On the other hand, when microneedles were loaded with nanocrystals, they were able to deliver high doses of hydrophobic drugs through the skin, locally and systemically, as we saw for the case of itraconazole and albendazole, respectively. One of the key points of this combination of technologies is the potential for long-acting drug delivery, which can be useful for the treatment of multiple diseases. So this is the end of the presentation. I would like to thank you all for your attention, and please do not hesitate to contact me if you need anything. My contact details are in this slide. Thank you very much.